Hi, I'm Melanie. Welcome to the Dan Lab. We get a lot of questions about how to collect metabolomic samples, so today we're going to talk about one aspect of collecting really good metabolomic data, which is to do the metabolite extractions while your cultures are in their growth conditions that you're testing, or as close as possible to that scenario. So here we have some bacteria growing for an experiment. Let's take a minute to zoom in on these.
Our culture is teeming with millions of living cells, and each cell has an active metabolism, where metabolites, represented here by the different colors, are constantly being converted and exchanged. Our goal is to isolate all of these metabolites to catch a snapshot of the metabolome at this moment under this specific growth condition. To collect the metabolome, we instantaneously lyse the cells and halt metabolism. Here, the colored dots in the petri dish represent the metabolites that were isolated by the extraction, and this is an ideal scenario, where our dish of extracted metabolites contains a good representation of all the metabolites that were present in our growing cells, as well as their relative abundances. However, if while our cells were still growing happily, they encountered an environmental change, for example, a change in temperature as the culture was removed from the incubator, bacteria respond rapidly by altering their metabolism. Then when we lyse cells and extract metabolites, the snapshot that we take is a picture of metabolism responding to the cold. This is not a good representation of all the metabolites and their relative abundances that were present in the growth conditions that we were testing.
To avoid compromising your metabolomic samples, do your metabolite extractions in the same conditions that you're growing your cells in to test. So in the warm room or in the anaerobic chamber, if that's where your cells are growing. If that's not possible, like in the case of a shaker like this, then we recommend setting up your workstation for metabolite extraction as close to the incubator as possible and removing cultures one at a time to do the extractions. That's all for today. Thanks for stopping by.